Every year, more than 1.5 million people develop cancer in the United States alone. Over 500,000 of them will succumb to the disease. Many types of tumors, such as lung cancer, are associated with changes at the genetic level. Understanding these gene mutations is the key to precisely identifying a disease and determining the best treatment. Next-generation sequencing technologies, or NGS, play an increasingly important part in this. They enable us to quickly and cheaply analyze even large amounts of genetic data, such as the complex genetic structures of a tumor. Leading institutes and hospitals already use sequencing data to diagnose rare diseases and to customize cancer treatments. However, there are still obstacles hindering the widespread adoption of NGS technologies in everyday clinical work. We are at the dawn of a new era in personalized healthcare. Our knowledge of the molecular mechanisms in cancer is growing, and these mechanisms can be triggered with targeted therapies. Given this, our task as pathologists is to analyze the tumor to determine which treatment can be effective and which not. This is becoming increasingly important with the development of new therapies and improved diagnostic technologies. However, while previously we were manually evaluating individual genetic mutations, today we want to evaluate a whole lot of them and do this quickly with a high sensitivity. This is only possible using high-throughput technologies that have a high level of automation, not only when it comes to DNA extraction, but also during the sequencing process and applied to an end-to-end bioinformatics pipeline for the precise analysis of data, identification of relevant mutations, and determination of the best therapy. Many existing NGS systems cannot meet these demands since they were designed for scientific research, but diagnostic laboratories have different needs. These needs can be met when each step of the process is harmonized from sample preparation to bioinformatic analysis, as with the NGS workflow solution QIAGEN is creating. The combination of instruments, QIAcube, QIAcube NGS, and GeneReader will automate the entire workflow and minimize manual interaction, reducing potential sources of errors. Along with specially adapted consumables, including QIAGEN gene panels for targeted enrichment of relevant DNA sections, the instruments will ensure top quality, precise results. At the same time, QIAGEN's NGS workflow will be highly flexible. The new GeneReader, for example, will be able to load up to 20 flow cells, with sequencing for each one carried out independently from the others. Even small sample volumes will be processed in tandem, improving lab efficiency and flexibility in clinical practice. The system will also support users in what is arguably the most important step of any analysis, transforming the raw sequencing data into valuable molecular insights. This is enabled through advanced bioinformatics systems that cover all stages, from processing and analyzing raw sequencing data through its interpretation to final reporting. Market-leading solutions from CLC Bio and Ingenuity Systems will enable the transformation of raw sequencing data into valuable molecular insights in no time at all. This can shorten the time needed to make a diagnosis, from the weeks or months it has taken up until now to a few days or hours. The answers to many health-related questions are hidden within our genome. We want our clients to be able to access these answers as quickly, reliably, and efficiently as possible. That is precisely why we have developed a complete system that is tailored to the needs of our clients in clinical research and diagnostics, and that encompasses all workflow steps from raw samples to valuable molecular insights. This year alone, more sequencing data will be collected than in the entire history of humans. And the amount of this data continues to grow rapidly. This promises to bring swift progress in research and clinical practice. However, this is just the beginning of the transformation, and its end is still far from foreseeable. QIAGEN, making improvements in life possible.